Thanks very much. Good morning, everyone, and many thanks to the organizers for the kind invitation to speak here. As you can see from the title of my talk, I'm going to talk about fast 2D NMR methods and their application to quantitative metabolomics and fluxomics. The main research topic of my research group is the development of quantitative NMR methods. So basically we are developing NMR methods, pulse sequences, while keeping in mind the strong requirements of analytical chemistry. And over the last few years we've been focusing quite a lot of quantitative multidimensional methods and on ultrafast 2D NMR. I will talk about it in a few slides. We are applying these methods to a variety of application fields. The historical one is isotopic analysis. I won't talk about it today. And during the last few years, we've been starting to be interested in quantitative metabolomics, and this is what I'm going to discuss today. So I guess that many people already know that this is the basic workflow of NMR metabolomics, which generally starts with recording the proton NMR spectra of a high number of samples. And as you can see, of course, these samples show very complex proton spectra with a lot of overlap between peaks. So because these are complex spectra, what you typically do is that you do some spectral decomposition called bucketing or binning. You basically cut the spectrum into pieces and you measure the signal, the quantity of signal that you have in each bucket. And then what people do is that they do some statistical analysis to obtain an efficient discrimination between those samples. And of course, if you do further statistical tests, you can try to determine which bucket is responsible for this discrimination. But this approach is of course very efficient, but it can be a little bit limited if you want to go further and if you want to go some targeted individual quantification of some metabolites. That may be difficult, as you just said before, because of overlap between peaks. And if you want to go further, well, there are several approaches, and the one we are developing is to use quantitative 2D NMR. So, for those of you who are not familiar with NMR, a 2D NMR spectrum looks like that, okay? And you should imagine that the peaks are coming out of the screen. Actually, a 2D NMR spectrum looks like that, but we generally represent it like this in a contour plot mode. And why using quantitative 2D NMR for quantitative analysis? Because if you look at this example of a simple metabolic mixture, when you have a lot of overlap in 1D, on the 2D spectrum the peaks are spread in 2 dimensions, and then the risk of overlap is much reduced. And in principle you can obtain a much better quantification with a better precision from these 2D spectra. Now this is not something which is evident, because 2D NMR is associated with a number of challenges. The first one, which is a drawback from the quantitative point of view, is that the 2D peak response is peak dependent, which means that the 2D peak volume is influenced by a number of factors which are not only concentration but also relaxation time, J couplings, etc. So you have a response factor which is different for each peak. It means that if you want to do quantitative 2D NMR, the first choice is to correct for all these factors, but that's almost an utopia. And it means that you have to do some calibration procedure, like for many other analytical techniques actually. And the second drawback of 2D NMR is the long experiment duration, because to record a 2D NMR spectrum you need to repeat a high number of 1D experiments, and between them you have to wait until the system is back to equilibrium. So this is going to create timetable constraints, of course, and it's also a drawback if you want to study unstable samples. And the drawback from the point of view of the analytical chemist is actually that long experiments are more sensitive to hardware instabilities in the course of time. And in 2D NMR this creates some kind of noise ridges which are known as T1 noise, and which are going to affect the quality of your quantitative procedure. So during the last 10 years there have been a number of groups who have been trying to develop relatively fast quantitative 2D NMR approaches. We've been working on Homonuclear 2D NMR. I will detail that on next slide. A lot of work has been done by the group of John Markley as well. They have been working on heteronuclear 2D NMR to quantify metabolites in plant extracts. So here is a typical scheme of what we are doing. We are working actually with biologists who are interested in understanding breast cancer metabolism. So these people are growing breast cancer cell lines at the lab, and we are extracting the metabolites from these breast cancer cell lines. So we are working on extracts. We know that this extraction procedure is quantitative and reproducible. And on these extracts, what we first do is that we record a 2D spectrum to identify the signals arising from the metabolites, from the main metabolites, and we identify those metabolites that we want to quantify. And once this is done, we do what is called a standard addition procedure, which is that we prepare a standard mixture containing all the metabolites that we want to quantify in a concentration that we know, and we spike the extract with such mixture, and by doing that you can have access to the initial concentration of the metabolites in the extract. And we've been able to do that on several breast cancer cell lines, and what you have actually in the end is absolute concentrations in the NMR tube, but they are normalized of course to the number of cells. And you can obtain that for different metabolites, and you can observe different significant differences between cell lines, which can be used of course by the biologist. Now I want to talk about ultrafast 2D NMR. In this context of being limited by the experimental time, as I said before, during the last 20 years or so, NMR spectroscopists have been trying to propose methods to go faster, to do faster 2D NMR analysis. I won't detail all of them, but the one we are working on is probably the most efficient in reducing the experimental time. It's called ultrafast 2D NMR, and it makes it possible to obtain a whole 2D spectrum in a fraction of a second, instead of a typical duration which is between a few tens of minutes to a few hours. This ultrafast spectroscopy has been proposed by this guy, Lucio Friedman, in 2002, more than 10 years ago. I won't go into the details of the NMR methods, but just to tell you the basic principle, if you want to do a conventional 2D NMR experiment, the typical sequence of events that you do is this one. Basically, you have to repeat a high number of 1D experiments on your sample while incrementing a delay which is called evolution period. And the problem comes from the fact that between all these 1D experiments, you have to wait until the system is back to equilibrium, and this can take several seconds. The key feature of ultrafast 2D NMR is that instead of repeating all these 1D experiments, you're going to perform all of them at the same time on different slices of the NMR tube. So all these experiments are done at the same time on different parts of the tube, and you can obtain in a single scan the data which is necessary to have your 2D spectrum. So of course it requires special NMR pulse sequences which are derived from imaging, and also special processing procedure. The interest of ultrafast 2D NMR is that depending on the kind of pulse sequence design that you are doing, you can in principle obtain any kind of 2D NMR correlation in a single scan, as illustrated by these recent examples from our lab. During the last 8 years, we've been trying to improve the performance of ultrafast 2D NMR to make it applicable to real samples, and in particular to metabolic samples. When we started working with ultrafast 2D NMR, it was in 2006, and as you can see, the quality of the spectra was quite poor, and we were limited to model samples in high concentrations. And during the last 8 years, we've been improving the sensitivity, the resolution, the spectral width of these approaches, the line shape. And we've also been putting a lot of efforts in improving the acquisition and processing procedure to make them as automated as possible. Because as you can imagine, if you want to use them for metabolomics, you need to have procedures which are reproducible and which are as automated as possible to analyze a large number of samples, of course. And as you can see here, we are now able to obtain 2D spectra whose quality is actually very close to the one of conventional 2D NMR spectra, but in a fraction of a second. This is a bit out of the subject, but I just wanted to tell you that these methods are very useful to study compounds whose constitution evolves in the course of time. And of course, these kind of tools are very useful to monitor fast reactions. With a Brazilian group, 2 years ago, we showed that we could use it to observe real-time organic reactions. This was a 2 hours time scale, and in 2 hours we were able to record, I don't know exactly, but probably 100 of HSQC spectra. and we could see the signal from the reactant, which is decreasing, the signal from the product, which is increasing, and the signal arising from the unstable reaction intermediate. So these methods are not only useful to follow kinetics, but also to identify reaction intermediates. Another situation where the sample is evolving in the course of time is when you have hyphenated techniques, and this is the case of coupling NMR with HPLC. What we've been able to do here is that we were working in an online mode, so it means that the analytes undergoing chromatographic separation are analyzed in real time in the NMR spectrometer, and of course if you want to do that by 2D NMR you need to be fast. So we showed that it could be done here on a mixture of 3 natural products, where we were able to record I think COSY spectra every 20 seconds, and to obtain the COSY spectra of the separate compounds at their respective retention time. So coming back to quantitative analysis, we tried to figure out whether we could use this ultrafast analysis for quantitative metabolomics. And then comes the main problem of ultrafast 2D NMR, which is the sensitivity limitation. Of course, you are spending only half a second on your sample, so you cannot expect to get the same sensitivity than what you have in 2 hours of acquisition. To give you, so the limit of quantification or limit of detection is difficult to give because it strongly depends on your hardware, but to give you an idea, on our 500 MHz spectrometer with a cryo probe, we had a limit of quantification for a 1-scan experiment, which was 10 millimolar. Now we have a 700 with a cryo probe, and it goes down to 1 millimolar in a single scan. But still, these concentrations are generally higher than what you have in biological extracts. So it means that if you want to do quantitative analysis, you may need a little bit of signal averaging. Okay, but then if we need signal averaging, you could tell me, why don't you come back to conventional 2D NMR? And actually, we ended up in comparing what I like to call long ultrafast 2D NMR with conventional 2D NMR. And as you can see here in the case of COSY, what you have here is a repeatability of the approach, in red for conventional COSY, in blue for this long ultrafast COSY for the same experiment time. I think here it was 10 minutes or 15 minutes. And as you can see, we can obtain a much better precision, a much better repeatability on the quantitative results when you use the long ultrafast approach. And the main reason for that is that these ultrafast experiments are much less sensitive to hardware instabilities in the course of time than their conventional counterparts. This is basically because if you look at conventional 2D NMR spectrum, you have this T1 noise, and in ultrafast 2D NMR you have approximately the same signal to noise in the 2 dimensions. So for the analytical chemist, it's also quite interesting. And we've been able to apply that to different breast cancer cell lines again, and to compare, well, here it's just a feasibility study for the moment, and we compared 3 breast cancer cell lines, extracts, and we were able to determine the major concentration of major metabolites, and to observe significant differences between cell lines. So now, of course, we have to work with biologists on a higher number of cell lines and so on. The last part of my talk is another application of these ultrafast 2D NMR procedures to the context of fluxomics. We have a very nice collaboration with the group of Jean-Charles Portet in Toulouse. Maybe some of you know him. Fluxomics aims at understanding metabolic fluxes in living organisms, and what these people do is that they feed living organisms such as bacteria with C13 labeled substrates, here C13 glucose, and they try to see where the C13 atoms end up in the final products of the metabolism. It means that these people need to be able to measure site-specific C13 enrichments, which are specific of a specific site in all the metabolites. In simple cases, this can be easily carried out by 1D proton NMR, because if you look at the proton NMR spectrum of a C13 enriched compound, you can see that you have the satellites here, which correspond to proton band to C13. You have the central line, which corresponds to proton band to C12. And if you measure the area under the satellites divided by the total area of the pattern, you can have access to the site-specific enrichment. But of course, when you go to biological mixture, this does not work anymore, because you end up with this kind of spectra where you have a lot of overlap, because you have a high number of metabolites, and because you have a high number of isotopomers for each metabolite. So these people in Toulouse, what they are doing is that they record 2D proton-proton spectra with C13 decoupling in one of the dimensions only. And as you can see here, you obtain, for each peak, you obtain 3 peaks, and if you extract a line from this 2D spectrum, you can have access to the site-specific isotopic pattern and measure the site-specific C13 enrichments. And I think it was 4 or 5 years ago, these people came to us and they said, okay, we have this approach, it works very well, but it takes a long time. It takes, I think in their condition, it was taking between 5 to 10 hours per spectrum. And because they needed a very high resolution. And they asked us, could you do it by ultrafast 2D NMR? So after some work and investigation, this is what we managed to do. We were able to record such an ultrafast 2D spectrum, so it's not purely ultrafast. I think there were 8 scans here in 40 seconds. We get this kind of spectrum obtained on a biomass hydrolysate from E. coli, and we were able to extract lines from this 2D spectrum and to access isotopic enrichments from this 2D spectrum. And the isotopic enrichment that we obtained with the ultrafast methods, actually we did 2 different ultrafast methods, are the same as those obtained by the conventional techniques, but the experiment duration was down to 40 seconds. Now, we were also wondering if we could go further, because if you look at this 2D spectrum, actually it's true also on the conventional one here, or on the ultrafast one there. If you have metabolites which are close to the others or close to the diagonal, you may still have overlap between peaks here due to the proton C13 splitting. So we thought that it would be nice to do it in 3D and to tilt these C13 proton enrichments in a third dimension that would be orthogonal to this 2D plane. So we wanted to do this kind of fast 3D approaches where you have proton chemical shift in 2 dimensions, proton C13 couplings in the third one. So if you read a plane from this 3D spectrum, you have a fully C13 decoupled proton COSY, so with limited overlap. And if you read a column perpendicular to this plane, you have access to the C13 isotopic enrichments. So we did that. So if you want to do it by conventional 3D NMR, that might take a long time, because 3D NMR is typically several hours to a day of acquisition. But we did it by mixing ultrafast 2D NMR with conventional 2D NMR. Basically, we obtain a 3D NMR spectrum in the time which is generally needed to get a 2D one. So we get it in 11 minutes here. And we also applied that to biological mixtures. So it's the same extract from E. coli. And you can see that the overlap is much limited here. And we were able to extract, it's not very clear here, columns perpendicular to this 2D plane, which is extracted from the 3D, and to measure the site-specific isotopic enrichments. And as you can see here, we were able to measure more enrichments than in 2D. There were enrichments that were not accessible in 2D due to overlap. And in some cases, the value is even better. Here we had a value in 2D which was not correct, and which is now correct in 3D. So I hope I've convinced you of the efficiency of these methods. If you want to try them out, you can contact me, and we have a web page where we provide some kind of implementation protocol with pulse sequences and so on. I'd like to finish by acknowledging my group of students and colleagues. I'm working a lot with this guy, Professor Serge Coca. We are in the same group, and I have very nice PhD students, Mirakan Patan, Renaud Boisseau, and Adrien Le Guenec, and so on. I'd like to thank my collaborators here, especially the group of Jean-Charles Portet in Toulouse for this nice fluxomic collaboration, and the funding sources, especially this starting one from the French National Research Agency. And I thank you for your attention. So actually what we do is we don't get the peak intensity, we get the peak volume. So we are using the integration tool which is present in the 2D software actually. We have been investigating different solutions. You have the choice here, either you can measure the volume, or you can read lines and obtain pseudo 1D spectra and measure the area under the peaks. But typically we are using the peak volumes. It requires a very standard NMR hardware. All what you need is a probe with gradients along the z-axis, which is typically the case of almost all probes now. So and it requires a standard gradient amplifier. So if your machine is less than 10 years old, there should be no problem with that.